Hi friends, today our topic is development of nervous system. The internal female reproductive organs consist of three parts: uterus, fallopian tubes and ovaries. Uterus in turn consists of fundus, body or corpus, cervix and cervical canal. The cervix protrudes into vagina. Fallopian tubes consist of mainly four parts. They are from medial to lateral, intramural, isthmus, ampulla and infundibulum. During the first week of development of the zygote into embryo and fetus, the nervous system development, that is the both central nervous system and peripheral nervous system, also starts. In the mid-menstrual cycle, when the Graafian follicle is fully matured, there is luteinizing hormone surge and it triggers the ovulation process. Once the ovum is released from the rupture of Graafian follicle, it is taken up by the ampulla region of fallopian tube. From here, ovum starts moving towards uterus. Ovum has completed first meiotic division by this time, but not the second meiotic division. Ovum is stuck at the metaphase of second meiotic division. If ovum doesn't receive sperm, it never gets fertilized and it never completes its second meiotic division. Rather, it will undergo death process, that is ovum can't live without sperm longer. If by chance sperm happens to be there and once sperm fuses with the ovum and the ovum gets fertilized, the ovum which is fertilized has got the genetic material from both ovum and sperm, which are known as female pronucleus and male pronucleus. These consist of 23 chromosomes each from both maternal side and paternal side. These nuclei fuse together to become a diploid nuclei with 46 chromosomes. The fertilized ovum thus called zygote. Zona pellucida remains intact covering the zygote, but the zygote starts cleavage. It first divides into two cell stage and then to four cell stage, which is called blastula. Blastula further divides and makes about 8 to 12 cell structure called morula, and the individual cells are known as blastomeres. The cells then make a mass at the center called inner cell mass, which subsequently forms the future embryo, and the cells which are arranged at the periphery are called outer cell mass, which provide the supporting tissue to the developing embryo by forming coverings on the body of embryo like amnion, chorion and also placenta. It provides material required for growth of embryo. Then the zona pellucida starts dissolving and a lot of fluid starts accumulating. Outer cell mass will remain the same, but the inner cell mass gets attached at a particular point. This is called blastocyst. The fluid filled cavity here is called blastocele. In this blastocyst, inner cell mass is called embryoblast and the outer cell mass is called trophoblast. Blast means cells which proliferate rapidly. Embryoblast means the cells which are rapidly proliferating and they are going to make embryo. Trophoblast means rapidly proliferating cells which have trophic or growth value. From the trophoblast, some finger-like process called villi emerge. The center of each villus is made up of cells with their nuclei intact, but the cells at the periphery fuse with each other and make a network called syncytium, with all their nuclei floating in the cytoplasm. The cells at the centers are collectively called cytotrophoblast, and the cells at the periphery are collectively called syncytiotrophoblasts. Now, blastocyst reaches the uterine cavity. The tips of the villi of the trophoblast release destructive enzymes at the point of their attachment with the endometrium of the uterus and dissolve the maternal endometrium. Thus, the blastocyst gets implanted in the endometrium at about 9th to 10th day. Maternal endometrium in the second half of menstrual cycle is mainly acted upon by progesterone, which is released by corpus luteum, the remnant of Graafian follicle, thus making endometrium soft, highly vascular and secretory with all the glands well developed, preparing it for the arrival of the fetus. The corpus luteum has a lifespan of about 10 to 12 days, and after that it degenerates and supply of progesterone is lost. If once progesterone is lost, endometrium is lost. Endometrium, the lining of endometrium along with some bleeding is passed, and this is called menstrual bleeding. If that female is pregnant, as blastocyst is implanted in the endometrium, she should not lose endometrium and menstrual cycle should not start, and hence there will be a missed period. The cytotrophoblast, which is the covering of developing embryo, releases a hormone called human chorionic gonadotropin, which is a hormone for maternal recognition of pregnancy. Primary role of this HCG is to keep corpus luteum functioning so that the corpus luteum continues to produce estrogen and progesterone, which are required for the growth of the baby. HCG begins to decrease when the placenta is developed enough. Thus, this is the first week of development where embryoblast is developed. After 8th day of conception, HCG may be detectable in serum, and at 10th day, HCG may be detectable in urine. Here, HCG is released by trophoblast and not embryoblast. In the second week of development, embryoblast will start developing. The inner cell mass will convert into two layers, which can be seen as a circular ball cut in the middle transversely into two parts. Here, cells are arranged like plates into two layers. This is called bilaminar disc. Upper layer is called epiblast and it consists of amniotic cavity. The lower layer is called hypoblast and it consists of primitive yolk sac. Both epiblast and hypoblast are held together by procaudal plate, which is shown in orange color with circular shape. The importance of this procaudal plate is it tells us which is the caudal end and which is the cephalic end. Procaudal plate is going to be future mouth of the baby, and this develops into head and neck, and the caudal end develops into lower part of the baby. As soon as epiblast and hypoblast develop, some cells at the center of epiblast degenerate and make a primitive streak with primitive node, which is shown in green color, longitudinal one. The cells at the side of these primitive node and primitive streak start proliferating inwards towards the primitive node and downwards, forming two layers under the surface of epiblast. Thus, three layers can be seen now. The uppermost layer is called ectoderm, middle layer is called mesoderm, and the lowermost layer is endoderm. Thus, the bilaminar disc has turned into trilaminar disc in the second week of development. The layer of cells towards the amniotic cavity is ectoderm, and the layer of cells towards the primitive yolk sac is endoderm, and the layer of cells in between these two layers is mesoderm. The cells which grow here down the primitive node and moving towards the procaudal plate form a collection of cells known as notochord. The third week of development, here the notochord has already appeared in the mesoderm. The notochord secretes growth factors which stimulate the differentiation of the overlying ectoderm into neuroectoderm, forming a thickened structure known as neural plate. The lateral edges of the neural plate then rise to form neural folds. The neural folds move towards each other and meet in the midline, fusing to form neural tube, precursor to the brain and spinal cord. During fusion of these neural folds, some cells in the folds migrate to form a distinct collection of cells known as neural crest cells. They give rise to parts of peripheral nervous system, and the formation of neural tube is known as neurulation and is achieved by the end of 4th week of development. The neural tube consists of anterior pore and posterior pore. On the 25th day of gastrulation, anterior neural pore closes, and on the 27th day of gastrulation, posterior neural pore closes. Defects in formation of neural tube are anencephaly and spina bifida. If folic acid is deficient in maternal blood during pregnancy, then the anterior pore and posterior pore, which are the cephalic ends and caudal ends respectively, may not close properly and may end up with diseases like anencephaly and spina bifida. Anencephaly results from failure of neural tube to close at the cephalic end, leading to partial absence of the brain and the skull. The lack of these crucial brain structures means that this is a lethal condition, and newborns with this congenital abnormality typically do not survive longer than a few hours or days after birth. Spina bifida results from incomplete closure of the neural tube at the caudal end, most commonly in the lumbar region. There are 3 main types of spina bifida: spina bifida occulta, meningocele or meningeal cyst, and myelomeningocele. Spina bifida occulta is the mildest form and is characterized by incomplete closure of the vertebra without protrusion of spinal cord. Most people with this form of spina bifida are unaware of having it, and its discovery is often incidental. Next, meningocele. It is the condition where the meninges protrude between the vertebra posteriorly but the spinal cord is not damaged. And the next one is meningomyelocele, which is the most severe form, where a portion of spinal cord remains unfused and protrudes posteriorly through an opening between the vertebra in a sac formed by the meninges. This is associated with severe disability. In the 5th week of development, swellings appear at the cranial end of the neural tube. 3 primitive vesicles appear first, and subsequently these develop into 5 secondary vesicles. These vesicles will give rise to all the structures of brain, cerebellum, and the ventricular system. The most cephalic one is called prosencephalon, which will convert into forebrain in the future. The middle one is called mesencephalon, which is the midbrain, and the inferior one is called rhombencephalon, which is the hindbrain. The CSF filled cavity throughout the neural tube will stretch to form ventricles in different parts of nervous system. And the remnants of notochord in the adult are represented by nucleus pulposus in the fibrous annulus ring of intervertebral disc. Coming to the secondary vesicles, the prosencephalon, some part of the prosencephalon rapidly grows and develops and extends, which is known to be called as telencephalon, and some part of it slowly grows and develops and remains at the center, and this is called diencephalon. Mesencephalon remains mesencephalon. It does not make any secondary vesicles. And the rhombencephalon makes two vesicles, metencephalon and myelencephalon. Metencephalon is the superior one and myelencephalon is the inferior one. On the posterior or backside of metencephalon, cerebellar hemispheres develop, and what remains in the front is called pons. And in the myelencephalon, there is medulla oblongata. The lateral ventricles are the cavities which are formed in the forebrain, that is telencephalon, and the third ventricle is formed in the diencephalon, and cerebral aqueduct can be seen in the midbrain, that is mesencephalon, and upper part of fourth ventricle is seen in the pons of rhombencephalon, and lower part of fourth ventricle can be seen in the medulla oblongata of myelencephalon of rhombencephalon. This image shows the primary vesicles prosencephalon, mesencephalon, rhombencephalon, secondary vesicles which are developed from it, and the parts derived from the prosencephalon, cerebral hemispheres and glober pallidus from the telencephalon, and from the diencephalon of prosencephalon, thalamus, epithalamus and hypothalamus are derived, and midbrain is from mesencephalon. Pons and cerebellum is from metencephalon of rhombencephalon, and medulla oblongata is from myelencephalon of rhombencephalon. Thank you, friends. For more videos, please share and subscribe.